discussing, there are very few facilities, at least in North America, right, that have these opportunities. I'm not even sure what it is internationally.
I only really know the Max Planck Institute. I'd be curious to see what some of the accelerator facilities in Europe have been capable of doing in other realms. I know the ISIS lab in Britain has participated in spin relaxation to study carbon not nanotubes, but they're called buckyballs essentially. If you have these nanospheres of carbon, you can use this is high energy physics, but you can use these high energy physics techniques to actually study their composition and whether their applicability for material science. But I think the field of biophysics has demonstrated that pretty much any tool, technique, or application that has been used to study materials, chemistry, or any other kind of solid state matter is just as applicable, if not even more applicable to the biological realm, with just a little bit of tweaking.
And what I think would be really relevant, James, is because ultimately, so James is a very unique researcher on the globe, looking at this transcription process and the biophysics principles with very unique facilities. And can you just describe for the students some of whom are going to read us in high schools elsewhere, or will eventually go to high school, or even our wonderful counselor and extraordinary individual, Cameron, who I'm going to convince to go into biological computing. My sensibility is, what was it like for you? Can you just tell them a little bit about your high school path, what you studied, then what you studied in college, and how it led to this opportunity? Because I think it's really valuable for them to understand that, because often you're in school and you have no idea that you lead to somebody like yourself looking at the code of life.
Yeah, boy, it began almost 10 years ago now. I was I grew up in New York City. I'm from Staten Island, and I went to Staten Island Tech. And I remember I got involved in something called the Urban Barcode Project. Essentially, you go, you study a little, you take a class on the study of genetics and phylogeny for 1 or 2 weeks, and then you're matched up with a professor who is interested in participating in this program. And as a high schooler, you have the opportunity to work under a professor's tutelage, or more likely a grad student's tutelage, to do research and collect data, and then you can enter these symposiums and present your work. It's all it was all very, very exciting. And I was studying the phylogeny of cryptic species. In nature, there are some species that closely resemble each other on a physiological scale, but their biochemistry is so different that they can't interbreed. They behave totally differently. So their outward appearance and their anatomy would suggest they're the same, but their chemistry suggests that they're completely different. And this is so as we were sort of transitioning more into the genetic age of categorizing animals through a phylogenetic process, I was looking into genes that we could use as markers to distinguish between species that would account for these cryptic species that are chemically very distinct from their otherwise identical cousins. So, and I was doing this on actually freshwater leeches, because they happen to have a lot of cryptic species within that taxa. So,
and this was high school.
This was in high school, and throughout my high school career, I ended up taking a physics class and falling in love with physics and how quantitative and precise it is. And I just love approaching problems from a first principle, sort of very grounded approach to understand a phenomenon from its core, essentially. So when I when I came into my undergrad when I came into undergrad at Columbia, I actually switched over and gave a number of different branches of physics a try. I started with solid state physics. I was doing research on a material called calcium ruthenate that could be used in electronics potentially. But then that got me sort of interested in the energy sector. So then the following year, I was involved in plasma physics. And plasma physics is pretty much, in this day and age, focused entirely on nuclear fusion and the acquisition of perfectly clean, renewable energy from nuclear fusion. And as I was doing that, I realized one of my specialties, and this is sort of going back to my background in a genetics lab, is working in a wet lab environment on very, very small and delicate systems. So I took a quick break from plasma to look into electrochemistry. And I was working in a battery development laboratory and we were looking at ways to make batteries safer with electrolytes that are less likely to explode, essentially. And that was I did a lot of really great work there, but the whole time I was thinking to myself, man, I learned so much about all these remarkable tools. Now, one of the things that I did in the battery development lab that led to this line of thinking was I did a collaboration project with a biochemistry lab, believe it or not. I was working on electrochemical cells, but they were working on real cells, and they used a technique called stimulated Raman scattering. There's like a diode, like a specific chip about the size of a smartphone at the time. They're bigger now, but smartphones, I mean, the chip is smaller, but the smartphones have gotten bigger for some reason, and this little chip cost about $400,000, and so I wasn't allowed to play with the machine all too much, but it was remarkable that we could look into these batteries and actually watch the gradient distribution of a specific chemical inside the battery change as we ran the battery to discharge and we started to recharge it. And we were able to observe phase transitions around the formation of dendrites, essentially. I'm not going to get into the technical details, but we were able to, using the tool that has been used for biochemistry, observe a phenomenon inside batteries that would otherwise be unobservable. And it sort of sparked an idea in my mind that by combining tools and techniques of multiple fields together, we can essentially by turning science by taking science and instead of looking at it from these three distinct pillars, biology, chemistry, and physics, by combining them together and using the tools that were applicable for one to study another, we can achieve really, really great and tremendous things. And that sort of brought me back to my love for the biological systems. And I realized then it is possible to study these fascinating, complex biological systems without having to sacrifice the precision, the first principles viewpoint that physics afforded me. So when it came time for me to look into PhD programs that I may be interested in, I realized, hey, this is kind of where I belong. Why don't I look into biophysics programs? And that led me to Cornell University, to this project that is on transcription, which is a topic that I'm very much interested in.
And James, yeah, I was going to say, James, what's your? So I was asking one of the graduate students yesterday who was talking about detection of cancer in a more effective way, right? So basically said, well, the physics are there, just like the electron and different microscopes are available here, but it's the utilization of those tools to actually ascertain plan for what you really need to look at, right? Sure. And it's really what it's achieving. So what is your goal, your fundamental driving purpose, if you would, at the end of the day? Because you're doing a lot of work in the lab, you're having to understand all these techniques. It can be laborious to some degree for you, but you have a goal at the end of the day. And what I call it is the ultimate goal.
Yeah.
I would say for the group here, what is your ultimate goal for yourself that you would like to achieve?
Well, with this research, I am interested in understanding at a comprehensive and fundamental basis the mechanism that is responsible for cellular differentiation that can then be applied to the I guess not yet existent industry of organ development. Essentially, you can by taking undifferentiated cells and selectively differentiating them into tissues that whatever tissue you desire, as long as you know the fundamental layout of the organ itself on a cell by cell basis, that can be recreated using modern techniques. We're just missing two pieces of information. That is how cells selectively differentiate themselves, so that we can attempt to replicate that process in vitro, and two, how these stem cells actually maintain themselves as they age. So cellular aging and cellular differentiation sort of come together in the fields of tissue and organ development in vitro. And I think understanding transcription is the most basic starting point to eventually reach that goal. And who knows? I hope to reach it in my lifetime, but it's and maybe I will. But I think Steve is absolutely right. Having a big overarching goal that you can tailor your research towards is a great idea, because you'll have life and opportunity will throw you will push you in different directions, and as long as you keep your eyes on what's really important to you and what's really interesting to you, you will get there. You will get to the point that you've set yourself.